Hi all. Today we are going to discuss about plasma membrane. That is last class we have discussed about what is a eukaryotic cell, prokaryotic cell, and we have discussed about plant cell, etc. Now we are going to study each and everything that is present inside the cell. So we are beginning from the first, that is plasma membrane. And you must know that you have studied about little bit about this in 8th standard. If you see this plasma membrane, you can understand one thing. This is a 3D diagram of plasma membrane. It was taken from a foreign author book in order to make it clear. If you see that plasma membrane is the boundary of a cell. If cell wall is absent, if cell wall is present, it will be having one more boundary out of it. It is a boundary that separates inside and the outside of the cell. That is inside material and outside material of the cell. And when we look this plasma membrane under the microscope, it seems that it is floating. And there are a lot of things. You can see many green branch like structures, some bean shaped structures, many such things. And inside of the cell, we can see some microfilaments, cytoskeletons, many things that is attached to the plasma membrane. That's the 3D diagram. And this is the another diagram which will be little more clear for the diagrammatic part. So if you see this plasma membrane, you can see something is attached, yellow balls, something like a dumbbell shape in both the sides, and we can see a globular protein, glycolipids, carbohydrates, cholesterol, integral protein, peripheral protein, channel protein, many things. So we will go a little bit about the history of plasma membrane. Scientists were knowing about this, that there is a membrane that separates between the inside and the outside of the cell, inside wall and outside wall of the cell. But they are not much sure about what is its structure. So in 1960s, a structure was described which was given by this person, that is the Singer and Nicholson. They give a structure model known as the fluid mosaic model, and till now this is the most accepted structure in the cytology part related to the plasma membrane. And if you see this plasma membrane is having lots and lots of functions, because since plasma membrane is the outermost boundary of a cell. So plasma membrane has to control everything, what is coming inside, what is going inside, pH, temperature, flow of nutrients, interaction between the cell. Each and everything has to be controlled by the plasma membrane. So plasma membrane is also known as semi-permeable membrane. Semi-permeable membrane, why we, because it allow some other things, some other compounds, sorry, not some other compound, it allows certain elements, certain molecules to come in. But plasma membrane is also called as selectively permeable. Sometimes they will select also what to come in, what not to go out, what not to come, no, what not to go in. Everything will be selected, that particular function also plasma membrane do. And apart from that, this plasma membrane is having a lot of receptors, because this is the plasma membrane, I said this is the boundary of a cell. Since it is a boundary of a cell, any signal that comes from outside, it has to pass through this plasma membrane. Since it has to pass through this plasma membrane, it required something on the surface which has to receive those signals and process it. We will study about that later. So these are the functions of plasma membrane. And when we come to the structure of the plasma membrane, We can see that this is a lipid bilayer model. Lipid bilayer means whatever the yellow beads, something like a dumbbell shape, what you see, it is made up of lipids. Lipids means, and lipid is also arranged in such a way with some other chemicals that one part is hydrophobic, that means water loving, and another part is hydrophilic, that is water hating. So the outside part is water loving and inside part is water hating. For example, if you see from this 3D diagram, you can see a pinkish orange color where it is written as microfilament cytoskeleton. There you can see this plasma membrane wall. That particular area is water loving and outside part is water hating. Why they are hating water? Because outside part there are lot of water is there. If they are water loving, more water will come inside. In order to avoid that, they keep water away. Whenever they want, they bring it in. And next of structure, if you see, there are some integral proteins. If you see these integral proteins, these integral proteins are the proteins that is present in between this in between this lipid bilayer. Since it is present in between the lipid bilayer, they serve as channels for the molecules to come in and go out. There are a lot of molecules such that which can pass like that. They need those things. And cholesterol and glycolipids, they give that water hating part, that is hydrophobic part, to the outer part of the thing. So this is the some of the basic structure which you have to know about this plasma membrane. Apart from this, and there is another question is possible to ask, why, if plasma membrane is having a structure like this, how they transport the materials in and out of the cell? So there are a lot of ways how they are transmitting the materials in and out of the cell. One of the most common way is diffusion. I will show the picture of this diffusion and osmosis after this slide. Diffusion means simply move from one part of the cell to the other according to the concentration gradient. That means wherever the water molecule concentration is high, from there to the other part. And whenever the process is diffusion, there is no need of energy. Remember one thing, anything that has to pass plasma membrane, it has to be in two conditions. Either it has to use energy or it should not use energy. Why they are using energy, why they are not using energy? There are some molecules are there which is required all the times. For that, the cell is not ready to waste the energy. That will be channeled across through a process of diffusion and osmosis. These two process takes place very commonly. If it has to run against a concentration gradient, they use ATP as energy and they will run those cycles. For example, for some of the few examples like sodium potassium pump, these are functioning on those by the use of ATP. So here we are going to discuss about osmosis. So what do you mean by osmosis? Osmosis is a special type of diffusion in which the solvent will move from the higher concentration to the lower concentration. For example, there are three types of solutions are there. One is isotonic, hypotonic, and hypertonic. Isotonic means if the solute part is equal in both the side, then it is known as isotonic solution. Hypertonic solution means one side of the solute, one side of the solution is less in solute and another side is either equal or more in solute. Hypertonic means outside, that means whatever the solution that is present outside the cell, it is very high. So each of this, either it is a plant cell or an animal cell, they have to undergo this process, because anytime the pH, the solvent, the solute concentration, etc., can change inside and out of the cell. So it is very important, and you must understand that how this osmosis is taking place. The part of osmosis which takes place in plants, that means not part, the osmosis that takes place in plants and in animals are different. That means it functions in different way. For example, plants, they require more water, and animals, they don't require much water. So for an animal cell, isotonic solutions will be better, and for plants, hypotonic solutions will be better. That means outside hypotonic, so the water from the outside will come inside. And if we keep an animal cell in hypotonic solution, the water will come inside and it will burst the cell. So I will show you with the diagram in the coming slide. So the in the in plant cell, that will not takes place. But if it is an hypertonic solution, again the cell will burst, because no, sorry, cell will not burst, the cell will shrink, and then finally it will burst off. So that is why whenever the some slugs and some leaves, when they die, finally after the water is gone out, they dies on the process of this. So I think you got it. After this, we'll move to cell wall. You know that plant cells, in the eukaryotic cell, plant cells have cell wall. Cell walls are very rigid and tough, and this is one of the most distinguishing character between an animal cell and a plant cell. Since cell wall, because the cell wall is very tough, this is one of the main reason why plant and plant-like organisms can survive in adverse conditions. Because if the cell wall is absent, the plasma membrane, there's a chance that the plasma membranes, either it will shrill or burst because of the change in the solute level inside and outside the environment. Since the since the cell wall is present, they give an extra mechanical support to the particular cell. And cell wall is porous also. if, because if the cell wall is not porous, there will be no movement of molecules in and outside of the cell. And this is one of the main reason, because cell wall is the main component that makes these leaves and other things very stable. And the cell wall is made up of a polysaccharide known as cellulose, hemicellulose. By these materials they are made up of. And that is why when we eat any raw food, it is very difficult for us to digest it. And many of the vegetables and plant products we cook so that the cell become very soft, and which is help us to digest all the, not all the raw, all not all the vegetables, but few vegetables we are eating and few vegetables we can digest. And next one is difference between, now we are moving enter into the cell. For that we have, since we are moving inside of the cell, we have to know the difference between three things: protoplasm, cytosol and cytoplasm. I have given a diagram there. It's very simple to remember. The term protoplasm and cytoplasm is depend upon the presence and absence of the nucleus. If the nucleus is absent, that particular area, that means the liquid part or a portion that is in between the nuclear membrane and the cell membrane, it is known as cytoplasm. If we are coining the term protoplasm, It means everything inside the nucleus, that inside the cell, not nucleus, inside the cell, which includes nucleus also. And if it is a cytosol, it is only the aqueous component of the cytoplasm. Now we are not discussing about a nucleus or some other things. Whenever we use the term cytosol, it means it is the aqueous part, not any other part. Whatever the things that is suspended inside the that liquid part, that is considered as cytosol. And there are few MCQ questions I have given down related to this turgidity, that means osmosis, and related to this protoplasm and cytoplasm. Can you do it? And if you have any doubts, revert back or put a comment in the comment box. I will get back to you. Thank you. This is a diagram that represents the tonicity. This was taken from net. Since the CBC has not given that diagram, only sentence part is there. So you have to understand it better. So if you see this, you can see that three solutions they have given: hypertonic, isotonic, hypertonic. And in one thing they have placed animal cell and one they have placed plant cell. So hypertonic solution means if the solution outside of the cell, they are less in solute concentration. So in animal cell, what will happen? The water moves in and it will be lysed. But in plant cell, it will become turgid. Turgid means it not look good. It is something like a fluffy nature. The original structure is lost. But in isotonic solution, sorry, turgid means it become very big, but it will not burst off. Sorry. Turgid means become very big. It will not burst off. That means it's because of the cell wall, it will not burst off. And isotonic solution means in isotonic solution where the solute concentration is equal in both the sides. And animal cell is very better, feels better in the isotonic solution. But if you keep a plant cell inside that, it will become flaccid. Flaccid means not looking good, fluffy in nature. And if you're keeping it in a hypertonic solution, the cell will shrill, that means dry, and finally it will burst off. And same thing will happen inside the plant cell. So related to this, I have given few questions down, MCQ questions. Answer it. And if you have any doubt related to this, please put in the comment section. I will revert back to you.